Good evening. Welcome to the 28th session of PSI online lecture series. I welcome you all for this session. And as you know, for the last four sessions, we are continuing the cardiovascular system, physiology of cardiovascular system. This is not really a replacement of your theory classes, but it is believed to be supplement your learning procedure and your learning quest. This session is not only live, it will be available. It is all the like all other sessions, it is available, it will be available on YouTube, so that both synchronous and asynchronous learning can be supported by this effort. Today we have Dr. Santanu Chaudhary with us. He is level 3 assistant professor in the Department of Life Sciences, Presidency University, Kolkata. He has completed his undergraduation and post-graduation from Calcutta in the subject physiology. Then he continued to PhD in molecular physiology. In his credit, there are plenty of things to be mentioned. At least few things I can't restrict myself to mention. Lieutenant Colonel N. Bose Scholarship Award to study abroad from University of Calcutta, and he has completed his PhD from the Miami University, USA, in molecular physiology. Then there are many other awards, and he has got trained in developmental biology, cellular and molecular biology, and cardiovascular development and disease program. He is currently in teaching with high level of research, and he has publications, 22 publications, and more to mention, he has some good amount of research project he has completed, and few are ongoing. They are really appreciable. He has completed projects from CSIR, DST, UGC, DBT, and currently DST project is running under him. So we are expecting a good session on cardiovascular system, a different aspect of cardiovascular system, where developmental part we will learn, and we will see how the research involved in that. Welcome, Dr. Chakraborty.
Thank you. Thank you for the nice introduction. Um, is it visible? The slides visible?
Not yet. I am putting that in the. Yes, now it is visible. You can click that hide part, stop sharing and hide.
Yes, yes.
Yeah,
Okay,
yeah,
okay,
that's great. That's great.
okay.
So,
Great.
uh, really, I welcome you, and I hope we will be having a good session, Dr. Chakraborty. And the session is yours.
Okay, thank you. Thank you again, Dr. Nak, for this nice introduction. So, as I joined Presidency in 2013, but since 2006, I'm working on various aspects of heart development and disease. But first, I would like to thank the organizers, especially Dr. Nayak and Dr. Vishas, for this invitation. So today, my title, it says a pulsatile bio-incubator, cells, signaling and beyond. So I will try to convince you the adult heart as a pulsatile bioincubator, where different kind of cardiac cells, signalings or affected signaling pathways, both during development and disease, can lead us to get new ideas for therapeutic intervention again a lot of different cardiovascular diseases. Now, to start with, as a developmental biologist in training, so the heart is the first pulsatile organ to function in the developing embryo. So as you can see here in human developmental stages, around day 15, we can see this cardiac crescent-like structure as it initiates the heart developmental process. Later, around after 7 days, around day 21, we can see the heart tube formation. At those stages, we can clearly see the different structures of substructures like the atria, ventricle, conotrungus. Later, around day 28, we can see this looped heart where we can see these two distinct regions. One is the conotrungus region and another one is the atrioventricular region. From these two regions, we can see the development of the two sets of valves, two atrioventricular valves, mitral and the tricuspid, and two semilunar valves, that are aortic and the pulmonary. And finally, out day 15 gestation, we can see these four chamber structures with proper growth and maturation. So this is a basic developmental process or cardiogenesis. Now, needless to say, the adult heart is a complex organ, as you can see here from this impression. So it consists of the four distinct chambers, two atria and the two ventricles, different arteries and veins, and two sets of valves, as I just mentioned. But importantly, like an incubator, this mature heart consists of several different cell types like the myocardial cells or cardiomyocytes, endothelial cells, smooth muscle cells, fibroblast, specialized conducting cells, and epicardial cells. All these different kind of cells give rise to the different kind of structures. For example, the endothelial cells give rise to the heart valves. The fibroblast plays important role, especially during the cardiac fibrosis of cardiac scarring post injury. Specialized conducting cells are important for maintaining the conduction systems of the heart, and epicardial cells, outermost lining of the heart, is also give rise to the different kind of cells. It's also known as the sort of cardiac progenitor cells. Now, today in my talk, I will mostly focus on the cardiomyocyte story. So, to start with, as I mentioned, so this is different kind of cells found in the adult heart. So today I will focus mostly on the cardiomyocytes, as I mentioned, and by cell number it constitutes about 25 to 40% by cell number, and by volume it's constitute about 70 to 85% in total adult volume. In contrast, the fibroblast constitute about 40 to 60% in terms of cell number, and volume also it varies depending on the age, species, location in the heart, and also during the cardiac aging. So all these different factors plays an important role to govern the different cell types found in the adult heart for maintaining both the normal and in disease condition. Now, today I will talk about one of the important cardiac or cardiogenic transcription factor, TBX20. It belongs to the TBX1 subfamily of the T-box genes. Importantly, mutations in human TBX20 have been associated with a wide array of congenital abnormalities like dilated cardiomyopathy, atrial septal defect, mitral valve disease. Importantly, upregulation of the TBX20 has been also reported in patients with the tetralogy of Fallot. This is a complex congenital disorders, mostly associated with the ventricular septal defects, stenosis, overriding aorta, like that. Now, very importantly, this TBX20 is expressed dynamically in different regions of the developing vertebrate heart, including cardiac chambers. Now, you can see here during development, as I mentioned, it's going through a loop stage. This TBX20 is strongly expressed in the developing heart. So, it tells us it's an important transcription factor which regulates the proper cardiovascular development. Now, back in 2005, at the same time, several labs for the first time reported the generation of the TBX20 knockout mouse model. But as you can see here, compared to the control heart, we are clearly see the outflow tract region, right and left ventricle. In the knockout mouse, we can see as a catastrophic effect on the heart development. and those TBX20 null embryos are lethal by embryonic day 10.5, which further indicate the importance of TBX20 in normal cardiovascular development. But on the other hand, due to this early embryonic lethality, we cannot use this knockout mouse model to study TBX20 function in later life, like in the fetal heart or even in the neonatal or in the adult animals. Now, to address these questions, we have, or our lab have generated a TBX20 gain of function mouse model where we are use a transgenic mouse model. So this is a construct of the simple transgene, which is the under control of the beta actin promoter, and with a floxed cat gene, and this is our gene of interest TBX20. Now, importantly, in the single transgenic animals, so this is non-functional. So it behaves like a normal control animals with normal expression of the TBX20. But when this single transgenic animals is mated with another transgenic lines called a Cree driver lines or transgenic driver lines, then we can specifically overexpress TBX20 on those pre-expressing cells to see the effect on the heart developmental status. So we ask the question, does this TBX20 function differently in early embryonic versus late fetal cardiomyocytes during development in vivo? Now we use the two different myocardia cardiomyocyte specific TBX20 gain of function mouse models. So the first one is the NKX 2.5 Cree. Why NKX 2.5? Because NKX 2.5 are known as the early cardiomyocytes and only present in the embryonic cardiomyocytes. So we use this NKX 2.5 Cree lines with our transgenic TBX20 transgenic mouse to specifically overexpress TBX20 in those NKX 2.5 positive cardiomyocytes and want to study the effect. And also, likewise, we also use the beta MHC3, which is active little late about embryonic day 14.5 to 17.5. So that is in fetal cardiomyocytes. This beta MHC3 is very active. So in that mouse model, when we use our TBX20 transgenic with this beta MHC3 lines, we can specifically overexpress TBX20 in the beta MHC3 positive cardiomyocytes. So in this way, we can use the same TBX20 transgenic mouse model with two different Cree lines to want to study the TBX20 function in both embryonic and in fetal cardiomyocytes. So first question is, does this TBX20 overexpression affect early chamber formation in vivo? So as you can see here, so this is STG, means that single transgenic or control animals, where we can see the expression of the normal TBX20, and this NKX 2.5 Cree and TBX20 or double transgenic means our experimental animals, where we can specifically overexpress TBX20 in those NKX 2.5 Cree expressing cells. Now, as expected, you can clearly see compared to the single transgenic, in the double transgenic, there is overexpression of the TBX20 as marked by this TBX20 antibody staining, which is expected. So similarly, we counted those numbers. So you can see this percentage of MF20 and TBX20 positive cardiomyocytes are significantly increased in the double transgenic animals compared to the single transgenic. And we also did Western blot. You can see the TBX20 protein is overexpressed in the double transgenic animals compared to the single transgenic. So this tells us we were successful in overexpressing TBX20 in the early embryonic cardiomyocytes. But very interestingly, these double transgenic embryos were lethal and they die between embryonic day 10.5 to 11.5. So as you can see here, so this is the double transgenic experimental animals, left lateral view, and this is the lateral view, and this is the control animal, single transgenic. We can clearly see there is a reduction or in the size of the developing heart compared to the normal or control single transgenic. So there is a, so when we overexpress TBX20 in NKX 2.5 overexpressing cardiomyocytes, there is a reduction in the overall heart development, as you can see here. Now below, then we cross section those looped heart. So this is from the control. We can clearly see this is a normal development of the looped heart. This is the left ventricle, right ventricle, outflow tract. But in the double transgenic animals, these are very smaller compared to the control ones or control litermates. Then we use MF20 positive staining to show that the, in terms of the cardiomyocytes, because MF20 stains the cardiomyocytes, they are more or less equivalent in number. But interestingly, when we use the twist one, which is the marker of a EMT process, because as I mentioned before, during development, the heart valve initiates around this embryonic day 9 or 10.5. You can clearly see this twist one, which is a promoter of this EMT process for heart development, is overexpressed more or less normally in both in experimental animals compared to the control. So which tells us overall this overexpression of the TBX20 results in a smaller heart, but with apparently normal EC formation or endocardial cushion formation, which is the progenitor of the heart valves. Now we want to address what is the signaling mechanisms or why it is embryonic lethal. So we first of all we did a staining against the proliferation marker of phosphohistone H3. We can clearly see there is a down regulation of the phosphohistone H3 staining in the double transgenic heart compared to the single transgenic, which is again quantified in here. The MF20 stains the cardiomyocytes and it is co-labeled with the phosphohistone H3. Now, interestingly, the P21 CYP1, so this is a cyclin-dependent kinase inhibitors, are significantly upregulated in the double transgenic overexpressing TBX20 compared to the controller. So, in contrast to the proliferating markers, the inhibitor of the cell cycles are increased in the double transgenic heart, as you can see here. Then we look through another cell cycle regulatory proteins like NMYC1, which is again down-regulated in the double transgenic heart compared to the control. Then we did a real-time or mRNA expression assays of different cell cycle regulators like the P27, cyclin 2, cyclin D1, cyclin E1, NMYC1. They are all down-regulated in the double transgenic over TBX20 overexpressing myocardium compared to the letermate control. But we saw the a negative regulator of the cell cycles, that is P21, is strongly upregulated in the double transgenic animals. Now, to address study further, we also did detected the downregulation of the ARC signaling in those TBX20 overexpressing cardiomyocytes in the double transgenic animals compared to the single transgenic or control myocardium. So this is again proved by the Western blot analysis. So this is the Western blot or expression of the phospho arc in the double transgenic heart compared to the single transgenics, and this is quantified. So there is a decrease in the expression of the active arc signaling in the double transgenic heart overexpressing TBX20. Now, next we want to see what happens if we express this TBX20 specifically at the later time point, so that is in the fetal heart. So for that, as I mentioned before, we used a different cree lines. Now we use the beta MEC cree lines, and so this is the data from this double transgenic heart and the single transgenic heart. So as you can see, there is an increased expression of the TBX20 antibody staining in the chamber myocardium compared to the single transgenic. This is expected because we are overexpressing TBX20. And then we also did some Western blot using three different time points: embryonic day 12.5, 14.5, and 17.5. We can see there is a increased expression of the TBX20 in the double transgenic myocardium compared to the single transgenic liter controls, and which is again quantified as you can see here. But the important thing is that in this case, these double transgenic embryos are viable. So this is complete contrast with the previous experiment. So when we overexpress TBX20 in late cardiomyocytes as marked by the beta image secrete, those embryos are viable and adults are also obtained. So I will talk about that later. But overall, we can see the increase thickness in the chamber myocardium, as you can see here. So this is the double transgenic heart, this is the single transgenic heart. You can clearly see there's an increase in the thickening of the ventricular myocardium in the double transgenic compared to the single transgenic, which is again quantified in here. As you can see, there is an increase in the compact myocardium thickness in the double transgenic animals. So consistent with this increased thickening of the ventricular myocardium, we then look for the proliferation, and as expected, we also detected the increased percentage of phosphohistone and MF20. So this is the cardiomyocyte specific proliferating cells are increased in the double transgenic heart overexpressing TBX20 compared to the control animals, which is again quantified here. And also, importantly, NMIC is a known direct downstream target for the TBX20. So TBX20 directly go and binds to the NMIC promoter and promotes its expression. So this is a direct regulation. So also you can clearly see there is increased expression of the NMIC in the double transgenic chamber myocardium compared to the control animals. And also I want to point out the increased thickness of the ventricular wall, which is again quantified in here. Now, to prove further, we did a chromatin immunoprecipitation assay using the single transgenic and double transgenic chamber myocardium from embryos, and you can clearly see around 17.5 embryonic heart, there is an increased binding of the TBX20 with the NMIC1, which tells us when you overexpress TBX20, then more TBX20 is able to bind and bind with the NMIC1 promoter to promote proliferation. So this is again proves that vitamin C3 mediated overexpression of the TBX20 promotes cardiomyocyte proliferation in contrast to the what just I have shown with the other prelines. Now we look for other different markers to again prove the increased cellular proliferation. So there is the increased expression of the BMP10, as you can see here, in the chamber myocardium compared to the control heart, which is again quantified, and at the mRNA level, and with the protein expression level, we did antibody staining with the phosphosmat 158, which mostly associated with the cellular proliferation. So as expected, we can see increased number of the MF20 and phosphosphat 158 positive cells in the chamber myocardium compared to the control litermate animals. And again, also we did the Western blot. We can clearly see the increased expression of the phosphosphat 158 in the protein lysate isolated from the double transgenic animals compared to the control animals. So which proves that this increased BMP10 and phosphosphat 158 signaling in fetal hearts overexpressing TBX20. Now, we did some of the other expression array. For example, we did the expression of the other T-box factors like the TBX2, TBX5, and also importantly some of the important markers for the conduction system like the connexin 40, connexin 43, and this is all the in situ hybridization. So because we made those individual probes specific for this different gene like TBX2, TBX5, and we did in situ hybridization to detect the expression of all these mRNAs. Now, interestingly, for the only TBX2, it looks like that it is downregulated in the chamber myocardium compared to the control. But other than that, the TBX5, connexin 40, connexin 43, these genes are strongly upregulated in the double transgenic heart compared to their litermate controls. And also we did the NPPA. So this marks the mature cardiomyocytes. We not able to see any significant difference between this expression. And all this data again quantified by the real-time PCR assays, as you can see here. So which tells us this vitamin C3 mediated overexpression of the TBX20 at least represses the TBX2 while inducing the expression of the TBX5, connexin 40 and 43 in differentiated cardiomyocytes. So now, if we can summarize this study, it is very interesting because the TBX20 is the same transcription factors in early embryonic cardiomyocytes when we overexpress TBX20, which downregulated proliferation with the inhibition of all the cyclins and ARC signalings, but promote the expression of the CDK inhibitors like the P21 CYP1. In contrast, when we use the same transcription factors in fetal cardiomyocytes around embryonic day 14 to 17.5, we saw increased proliferation supported by the increased BMP SMAT signaling, increased expression of the TBX5, connexin 40 and 43 with downregulation of the TBX2. So which kind of tells us that TBX20 is definitely play a very important role, and the important thing is that maybe the precise level of the TBX20 transcription factor can regulate this differential function in embryonic cardiomyocytes versus these fetal cardiomyocytes. Now, as I mentioned, these embryonic cardiomyocytes and in fetal cardiomyocytes are very proliferative, right? And different signaling pathways can regulate this cardiomyocyte proliferation as showed by different groups, including ours. But very importantly, right after birth, in the neonatal or even in the adult, there is hardly any proliferating cardiomyocytes. So there is the very few proliferating cardiomyocytes are only able to we can find in the adult heart. So this tells us that maybe when we overexpress TBX20 in our transgenic system, maybe it can promote the adult cardiomyocyte proliferation, which is very important, at least in the for disease pathology. Now, apparently, we not able to detect any overt cardiac abnormalities or hypertrophy in the TBX20 overexpressing adult hearts in vivo. As you can see here, compared to the single transgenic, the double transgenic heart, there is no significant difference between the heart weight to body weight ratio. Why it's important? Because normally, if there is an increase in the heart weight to body weight ratio, which indicates some sort of cardiac injury or cardiac insult, so at least in these double transgenic animals, we not able to detect any overt cardiac phenotypes. Now, very surprisingly, this DBX20 overexpression promotes proliferation and also results in decreased cardiomyocyte cell size in vivo. So, this is the cell size experiment we did with the WGA immunostaining, which actually marks the cell surface. And you can see in the DTG heart, these individual cell size are very small compared to the control heart, which is again quantified as you can see here. And also, as expected, we able to detect proliferating cardiomyocytes in this double transgenic heart as marked by the alpha actinin and phosphohistone H3. Alpha actinin marks the cardiomyocyte and phosphohistone H3 marks the proliferating cell. So, which indicate that there is a significant increase in the proliferating cardiomyocytes in even in the adult heart compared to the control heart, as you can see here. Now, not only that, we also able to isolate those individual cardiomyocytes from those animals. So, as you can see here, in the double transgenic animals, we able to isolate those small cardiomyocytes, and mostly those cardiomyocytes are mononucleated. This is very interesting because normally in the adult heart, most of the cardiomyocytes are binucleated or multinucleated because right after birth, after 1 or 2 cycles of proliferation, normally the cardiomyocytes exit the cell cycles, and usually it follows a 1 round or 2 round of uh, karyokinesis without any cytokinesis that results in multinucleated cardiomyocytes. But very interestingly, when we overexpress TBX20 in the beta MHC pre overexpressing cardiomyocytes, we able to see an increased percentage of the mononucleated cardiomyocytes, as you can see here, compared to the normal heart. In the normal heart, you can see mostly those cardiomyocytes are binucleated and also larger in size. So this is very interesting for us. So it tells us if we able to overexpress TBX20 in the adult heart, we can actually able to initiate the formation of the small mononucleated cardiomyocytes indicative of increased cell cycle activity and increased proliferation. And not only proliferation marker, we also did aurora B kinase, which marks the cytokinesis. We also able to see the cytokinesis markers in the cardiomyocytes isolated from the single transgenic animals. And this data is also quantified in here. So very interestingly, we able to show increased cell cycle activity and percentage of the mononucleated cardiomyocytes in double transgenic adult hearts. Not only that, normally in the adult heart, as you can see here, the P16-INC4A, which is the cell senescence marker, are strongly upregulated, but in our cardiomyocyte isolated from the double transgenic, we're not able to see those cell senescence marker, all those isolated cardiomyocytes, and also those early fetal markers like the beta MHC, SS, TNI. This is known as the fetal early fetal markers are also strongly expressed in those cardiomyocyte isolated from the double transgenic adult heart compared to the control cardiomyocytes, as you can see here. And we also did a Western blot to further confirm this finding, but increasingly, this increased expression of the fetal cardiac genes and decreased expression of the senescence markers are detected in the double transgenic heart overexpressing TBX20. Then we also look for the different signaling pathways downstream of this proliferation pathway. So we did again the phosphosphat 158 signaling. As you can see here in the double transgenic cardiomyocytes, we able to see more phosphosphat 158 and alpha actinin positive cells, which indicate the increased number of the SMAT signaling in those cardiomyocyte overexpressing TBX20, and also not in vivo but also we isolate cardiomyocytes from the double transgenic and single transgenic animals, and you can clearly see again the phospho 158 positive nuclei in this double transgenic isolated cardiomyocytes, and also the beta catenin are also strongly upregulated in the double transgenic cardiomyocytes, which indicate there is increased cell cycle activity in even in the adult heart because of the overexpression of the TBX20. Then we also did a lot of Western blot or the assay or at the protein level. So this is just the quantification of all those data. You can clearly see there is expression of the TBX20, NMIC, BMP2, SMAD, all are upregulated in the double transgenic heart, along with the phospho-3 kinase, AKT pathway, GSK3 beta, and beta catenin. These are all upregulated in the double transgenic heart overexpressing TBX20. Now we want to see if this overexpressing adult cardiomyocytes are more proliferative. Then if we injure those TBX20 overexpressing heart by a myocardial infarction or ischemia reperfusion injury, if those hearts are protected or not. So as expected, you can clearly see. So this is to control hearts. It's just vitamin C3, and this is the single transgenic, and this is our experimental heart. As you can see here, this is a CCTBX20 and vitamin C3. So this is the vitamin C3 overexpressing experimental heart, and this is the TTC stained heart sections or heart fragments. So this white tissue indicates the fibrosis or the dead tissue, and you can see this is very much reduced in the double transgenic heart overexpressing TBX20 compared to the single transgenic control hearts. This is again quantified in here. Area at risk over ventricle is almost same for all these three different groups, but significantly, the actual infarct size or the injured area in the chamber myocardium is much more decreased in the double transgenic animals overexpressing TBX20. So this is the very interesting data for us, which tells us this TBX20 overexpressing adult hearts are kind of protected against this ischemia reperfusion injury in vivo, which kind of a mimic of the myocardial infarction or heart attack in adults. So overall, we kind of summarize what I have shown. In adult mice, this increased TBX20 expression results in an increased percentage of the small mononucleated cardiomyocytes, which is very interesting because normally adult cardiomyocytes are multinucleated and larger in size and non-proliferative. And these isolated cardiomyocytes from the double transgenic heart are also marked by persistent expression of the fetal cardiac gene and decreased expression of the senescence markers without any cardiac hypertrophy. And moreover, these TBX20 overexpressing adult hearts are protected against this injury induction in vivo. So this is, then we came up with a proposed model. So as you can see here, the overexpression of the TBX20 along with this BMP SPAD 158 pathway along with PI3, AKT, GSK3 beta and beta catenin pathway can drive even the adult cardiomyocyte proliferation in the at least in the mouse model. So this is very important because this is one of the major problem after myocardial infarction. Now I am switching my gear. On a darker side, we all know that heart failure is a major global epidemic, and it is a leading cause for global morbidity and mortality. And over 20 million people diagnosed with at least first-time heart failure across the world. And these numbers are also very alarming, as you can see here. In 2004, about more than 1.4 million reported deaths in more than 6.7 million people hospitalized due to these several cardiovascular disorders. And these values are also estimated to be more than 2.1 and 10.9 million respectively by the year 2021. And also, the average cost per heart related insurance claim has also increased significantly from 4 to 5 lakh in 1920 to around 12 to 15 lakhs in 23-24. So this is not only uh, create a physiological burden and also economic burden for the patient population, also the families. Now, here, when we talk about the heart failure, so this is kind of seen in the autopsy samples from the heart failure patient. So this is called the cardiac scarring or cardiac fibrosis. So they again, which again marked by the increased hypertrophic response, increased fibrosis, increased cellular death, and obviously reduced level of cellular proliferation. Now, what happens during myocardial infarction and LV remodeling? So, normally after myocardial infarction, we can easily differentiate three distinct zones in the heart. One is called the infarct zone. So, this is the zone of actual injury where most of the cardiomyocytes are necrotic or apoptotic in cells. Then AR, this area at risk, where most of the cells are undergo distress, and then the healthy or remote zone where we can always find some kind of normal cardiomyocytes. Now here you can see, so this is the normal heart right before the myocardial infarction or occlusion of the coronary artery, which results in hypoxia related injury, then infiltration of the different macrophage and granulatory phase early remodeling, then followed through late remodeling, and ultimately we can see architectural and functional changes in the after myocardial infarction or heart attack, right? So here you can see most of the viable cardiomyocytes are lost or replaced by the cardiac fibrosis or cardiac scarring compared to the, as you can see, are healthy cardiomyocytes in the normal adult animals. So this is known as the post myocardial repair and the LV remodeling. So therefore, this type of injury can be easily done in the laboratory based model system. So this is injured adult rat heart, as you can see here, after cardiac injury. So where we can see the extensive and dysregulated expression of the collagen, as you can see here by the Mason trichrome staining, compared to the control heart where we cannot see any fibrosis. So basically, fibrosis can be defined as an extensive and dysregulated production and deposition of the mature collagen fibers throughout the chamber myocardium, which results in the severely compromised or functional insufficiency of the adult heart. Now, in this respect, in disease pathology, now I will talk about another molecule. It's called the Adam TS4. So it's a aggrecanase 1, belongs to a disintegrin and metalloprotease with thrombospondin motifs family of the secreted ECM proteins. It's an important extracellular matrix protein, and before our paper or research, it has been implicated in proteoglycan degradation, therefore pivotal importance in the tissue degradation and remodeling. Why it's important? Because I just mentioned, after myocardial infarction or heart attack, there is a massive remodeling in the chamber myocardium. Also, this Adam TS4 is associated with the pathology of several invasive tumors, including the glioblastomas. Moreover, the systemic deletion of this in mice associated with the increased osteoarthritis susceptibility, which again tells us its Adam TS4 is also already associated with the tissue remodeling, especially during injury or in pathology. Now, very interestingly, when we did this myocardial infarcted or generate this myocardial infarcted mouse model, for the first time we able to see there is upregulation of this RMTS4, this ECM regulators throughout the infract zone, border zone, and even the remote zone of the chamber myocardium. So this is for the first time we reported this expression of these RMTS4 ECM proteins after myocardial infarction or heart attack in mouse model. And consistent with this data, we also show this the expression of the periostin, which actually marks the cardiac fibroblast or fibrosis process. We can clearly see increased expression of this periostin along this infract, border, and remote zone of the injured myocardium. So this tells us this RMTS4 is reactivation occurs after myocardial infarction in vivo. Now, next we want to study what is the basic developmental expression pattern of the Adam TS4. This is also very interesting, as you can see here. So this is done in the mouse embryos. So this is the embryonic day 10.5. So this is the developing heart, as you can see here, 12.5, 14.5, 18.5. We can clearly see this Adam TS4 expression throughout the chamber myocardium as depicted by the Adam TS4 specific antibody, and this blue color indicates the nuclei. Now, very interestingly, in the adult heart, this expression is gone. So this is very interesting. That tells us maybe this RMTS4 is strongly expressed during normal developmental remodeling. We can call it physiological remodeling, and then its expression is repressed. Even in the normal adult, we cannot able to see the strong expression of the RMTS4. But as I mentioned before, but after injury, this RMTS4 is again reactivated as a important regulator to for pathological remodeling. So then we also did some co-localization study to show that this is co-localized with the MF20, so cardiomyocyte specific MF20, also in the embryonic time point and also in the adult heart after injury. We can clearly see this RMTS4 expression is co-localized with the cardiomyocyte marker MF20 as marked by these arrowheads, which tells us this RMTS4 reactivation in adult cardiomyocytes following injury in vivo. So this is very interesting for us. So maybe we can use this as a diagnostic biomarker after adult cardiac injury. So we also able to get hold of some human samples or human blood samples from these different patient populations. So you can see, so this is from the dilated cardiomyopathy, this is the anterior wall myocardial infarction, this is the control samples, interior wall myocardial infarction, again anterior wall myocardial infarction. So all these patient serum samples we able to detect the expression of the RMTS4 in their blood serum. As I mentioned, because it's a ECM protein, it can be easily detected in the systemic circulation. So this is a great advantage for us. So we can use this and able to show that there is an increased upregulation of the RMTS4 in the in patient populations having different types of cardiac ailments. And also along with RMTS4, we also able to detect the alpha SMA, which also clearly marks the cardiac fibrosis process. So alpha SMA is also activated in all those different patient populations. So this data is also quantified, as you can see here. So overall, this tells us that we can use this RMTS4 as a novel cardiac injury biomarker. As a result of that, we can say that if we somehow able to inhibit this RMTS4 activity right or before myocardial infarction, we can somehow able to reduce the burden of extensive or dysregulated cardiac fibrosis, which eventually can lead us to the better cardiac functions in patients after myocardial infarction. Now, to do that, so this is the last part of my talk. Interestingly, back in 2018, a group for the first time reported the availability of this cardiac targeting peptide. This is a small, about 12 amino acid small sequence of these cardiac targeting peptides, and this peptide can directly, if you inject it in the systemic circulation, it can go and directly go to the chamber myocardium. So this is called the cardiac targeting peptide. Now with the discovery of that, we able to find out a way if we can able to target this RMTS4 specific inhibitor specifically in the heart to able to reduce the burden of this cardiac fibrosis process. So what we did to initiate the experiment, we also able to attach the cyanin 3 fluorophore conjugated with the CTP, as you can see here. So this is the cyanin 3 probe attached with the CTP, and as a very preliminary experiment, so we did so this is the 3D tomographic imaging of the adult male rat injected with that cyanin 3 label CTP in the through tailvein injection, and we can clearly see detect the its presence in the heart region in the adult rat. So this is very exciting for us because so there is a inhibitor known as the compound 10 or 10% polysulfide that can specifically inhibit this RMTS4. So we are in a process now able to use this CTP targeting peptide. If we able to design that inhibitor and tag with this cardiac targeting peptide, maybe we can able to deliver that in the at least in the adult male rat and see what happens after myocardial infarction or after cardiac injury. And our hypothesis is that if we able to inhibit this RMTS4, we can definitely block this extensive and dysregulated cardiac fibrosis process, and maybe this lead us to better functional output in those adult injured rat model. So then the overall significance and impact. So we, for the first time, identified RMTS4 as a novel biomarker of adult cardiac injury under stress conditions. We detected a strong expression of the RMTS4 protein in the developing chamber myocardium compared to very restricted expression in the adult murine hearts. Moreover, these provide us a better knowledge to exploit these developmental gene reactivation mechanisms in the treatment of the adult cardiovascular disease in future, because this is true for both TBX20 and RMTS4. Because we know this TBX20 as well as RMTS4 is very important developmentally, one being a transcription factor and other is involved in the normal physiological remodeling during cardiogenesis. But after injury, this sort of developmental factors gets reactivated, and maybe this we can use that strategy to somehow, if we can able to dysregulate their expression, maybe it's a better way. We can use this as a therapeutic intervention for better cardiac output. And overall, again, we validated these human clinical samples and demonstrated the expression of RMTS4 in patients with cardiac ailments. And prolonged expansion of ECM could lead to extensive fibrosis, as I just mentioned about the extensive and dysregulated cardiac fibrosis, myocardial stiffness, and most of the time may turn fatal and results in the immediate heart failure. So, inhibition of these MMPs or RMTS4 could be a possible targets to reduce fibrosis induction post cardiac injury. And availability of such inhibitors, as I mentioned, like pentosan polysulfate or compound 10, that can be used to specifically inhibit RMTS4 using this cardiac targeting peptide strategy against this cardiac fibrosis process in clinics. So with that, I want to thank all my laboratory members, current and past members, and my research collaborators, also from my postdoc mentor from Cincinnati Children's Hospitals, and my collaborators from the Jadavpur University, and the patient samples. For that, I want to acknowledge Dr. Dipankar Mukhopadhyay from SSKM Hospital, Kolkata, and my presidency collaborator, Dr. Devrani Mitro. But not the least, all the funding agencies. Without their generous funding, I was not able to generate this kind of data. So with that, I want to thank all of you for your patient hearing, and I am sure that I able to give you an idea that maybe now we can call adult heart as a pulsatile bioincubator with all of different kind of cells and signalings and beyond, so that we can use this information, both developmental and disease information, for better therapeutic strategy. So thank you very much.
Dr. Chakraborty, uh,
Thank you. Thank you.
very, very good presentation. But however, I'm really afraid that our students, whether they can appreciate the level, it's a very good work, but
Mhm.
whether they can really appreciate that level or not, that's a question. However, I am sure one thing, that they will be stimulated, they will be they will find it more interesting now to read the cardiovascular system.
Right, and at
How
least what are the different ways we can go for it, like the proliferation, remodeling. So this is the very common after effects of like heart attack. So there is a
yes,
way, at
yes,
least
yes.
we can at least approach our research in that direction.
Exactly, exactly. From the pathology, physiology to pathology, how we can go, and how we can bring back,
Right,
if at least we can think of bringing it back to physiology,
right.
that's a really very good. And I, from on behalf of the society and on behalf of the audience, I thank you for
Thank you, thank you.
giving this nice lecture. I have just maybe a novice way of asking few things.
No, no, sure,
I'm
sure.
not able to resist myself to ask a question. Like, as you have mentioned that there are remodeling after the infarction, there are remodeling, and
Right,
that is virtually pathological remodeling,
right.
and there it is happening that the ventricular volume, left ventricular volume is increasing,
Hmm.
and that elasticity or that contractility part, physiological part, that is not
Right,
being maintained completely.
right, right,
What I would like to know, that in case of aging process, what happens? Like with aging,
right.
are we going towards pathology, or there is any different mechanism?
Right. I think the aging, with aging, I think there is a definitely loss of cardiomyocytes, but again, those cardiomyocytes are replaced by the cardiac scarring or fibrosis, but in a limited sense. But after injury, so there is a loss of large number of cardiomyocytes. So there is a kind of like a void space. So then there is extensive and regulation or deposition of the collagen fibers, and though in that case there is a replacement of like 30 to 35% of cardiomyocytes, and that results in the cardiac insufficiency. But with aging, the loss is kind of restricted. So but in aging process, if we challenge the heart, maybe if we somehow challenge the heart, they are more prone towards heart failure, definitely. But
Okay.
the important thing is that the cardiac fibrosis is often called like a necessary evil, so we cannot get rid of it, because after myocardial infarction, so there is a definite loss of cardiomyocytes, and those places should be replaced by the fibrosis. Otherwise, there is immediate rupture of the cardiac wall.
No,
You have that, but
that's
we need to
very
control that
sure.
process. Yeah,
Like in any other wound healing process,
right, exactly,
fibrostic fibers, they will be appearing, and they will do that process,
right,
injury, that healing process. And
right,
during healing process, you get some scar, and that type of
right,
scar it is.
right.
So is it possible, like, see, the loss in myocardium is controlled in case of aging? Can we
Yeah, yeah,
identify
yeah,
the,
yeah, that, yeah, the
like,
people are working on that, so they are trying to find out that I talk about this TBX20. So it looks like the TBX20 also plays a role in like the cellular senescence. So in the adult heart, most of the cardiomyocytes are either terminally differentiated or undergoing cellular senescence. So maybe if we can modulate the expression of all these transcription factors, then maybe we can get a better output. But it's possible. But the main problem in cardiovascular, how to deliver those targets, is very very difficult, because for transcription factor we cannot deliver to systemic circulation. For ECM proteins we can do that, but for transcription factor we cannot do that. So that's a major bottleneck, how to deliver those factors, proteins directly into the chamber myocardium.
so in case of, say, if we know the process of senescence, or if we know the process of myocardial restriction, loss restriction,
Mhm.
that can be helpful for us to identify the process, as well as to like heal the process, if
Right.
we can.
Now
And
I think, yes, now we are thinking about, so it looks like after any sort of injury there is a certain reactivation of the developmental genes. So if there is a way we can upregulate those developmental genes more, like normally what happens after injury, then there is a way we can generate more proliferating cardiomyocytes, maybe more tissue homeostasis. That is possible, but we need to find out the key regulator which who actually drive those developmental genes. Normally those are silent in the adult heart. So is there a way we can drive those reactivation program, then it is possible to generate more cardiomyocytes in the adult heart.
how about the other factors, like, say, atherosclerosis,
Yes,
or
yes,
other
yes, yes.
dyslipidemia,
Right,
or
right.
oxidative
Yeah,
stress?
something, yeah. So I thought I will also show some data, but it looks like too much data in here. So I
Okay.
also looks like the lifestyle disorders like the obesity or diabetes. These two factors specifically, when we induce these two factors in, at least in the animal model, looks like we are getting atherosclerotic plaque, or even in the muscles we able to get calcification. This is very interesting, because normally when we talk about the calcification, it looks like most of the time the calcification is affected only in the aortic valves, the aortic valvular cusp. But now looks like, when especially in obese condition or in diabetes condition, the muscles are also getting calcified. So it looks like the main regulator in this case is the inflammation. The cytokines or macrophage, those cells from the systemic circulation get into the chamber myocardium, and then they will differentiate different kind of cells into the osteoblast like cells, and they are the responsible for generating those specific loci, like the calcific loci throughout the chamber myocardium. So this is looks like the actual player in this case is the inflammation, I think. So now there is another field like coming big time like cardiovascular immunology. So look like all these injury insults are kind of induce these inflammatory cytokines, and they comes and they will modify the resident cells and generate a lot of different, like the calcific foci or fibroblast. They can trans differentiate all those different cells.
Okay. How about this, like antioxidants? They are coming very commonly.
Exactly. Yeah. Yeah. Yeah.
Antioxidants and other
Right.
supplements. They say
Supplements.
that these are healthy diets, or
Exactly.
heart, keeping the heart healthy, or
Right.
some exercise, or
Exercise.
say some modification of lifestyle.
Right.
How these are like going to help? Like, are they really going to switch on some specific molecular target, or gene target, so that
I
it
think,
can
yeah,
give
I
us
think the
a
what
benefit?
you mentioned, that the healthy lifestyle or healthy things probably switch up the developmental gene program. So I think those will activate those developmental gene program, and you are getting good factors, and you are repressing those bad factors, and maybe that's the reason you are getting healthy cardiomyocytes. And same thing for the different plant extract, like one of my students is working on the ficetine. It's a plant flavonoid. Looks like it this is antioxidant. When you gave those ficetine or flavonoid to the injured rat model, they are getting better results. So we are getting better results, better cardiac function. So definitely those antioxidants and healthy lifestyles definitely helps to get rid of all this, like the bad pathological remodeling.
Just maybe it's becoming lengthy. Still,
No, no, it's
I
okay.
feel.
I'm also enjoying. Yeah.
No, what happened? What happened to those, say, uh, concentric or eccentric hypertrophy of heart? It
Right.
happens sometimes
Right.
pathological, sometimes
Uh-huh.
in
Physiological.
athletes. In
Athlete.
athletes,
Yeah.
physiological hypertrophy of heart, it
Right.
happens. So,
So it yeah, in both
how
the cases,
you?
I think, right, in both the cases, both in the concentric or eccentric, in it looks like in when you're doing a physiological exercise, again goes back to a normal thing. But if it's in a pathological, or if you're doing a bad lifestyle choices, if you are a chain smoker, or do you drink alcohols or bad lipid based foods, then definitely it's going to a pathological concentric or eccentric hypertrophy, and eventually it goes to the dilated cardiomyopathy and eventually heart failure. But that is not happening when you are doing a pathological, I mean physiological hypertrophy. That's fine. They are more flexible, then you are getting back your normal cardiac homeostatic or cardiac output. But if you're doing a pathology, definitely it leads to the dilated and then heart failure.
My question is, like, where can we find the switch? Like, one side of it is bad, one side of it is good. How we can identify that switch? Like,
Maybe those
physiology
markers, those
is.
biomarkers, maybe those biomarkers can, maybe it's difficult, maybe you need 3 or 4 different biomarkers, because if there is a cardiomyocyte death, then you will get one sort of signals. If there is cardiomyocyte proliferation, then you will get another set of signals. So by routine checking up of the different biomarkers, you can clearly see if you're going a pathological or physiological uh, hypertrophy.
Anyway, thank you. I enjoyed it, and I hope all audience also enjoyed it. And thank you very much for your time,
Thank
and
you, and also
for
thank you
your.
for this invitation. I'm really liking this, and I'm again happy. If you need me for any other occasion, please let me know. I'm more happy to visit you guys, or again online, no problem.
Okay. Thank you very
Thank
much.
thank you very much. So I'm leaving. Okay.
Yeah, yeah. Please. Thank you.
Thank you.